Alright, so we're going to talk about the scientific method, and I'm sure this is something that most of you are probably already familiar with, so hopefully this will be a little bit of a review. We might go a little bit more in depth, but the first question that we need to answer is, what is science and how is science done? Science is our way of understanding the natural world. It's based on our five senses, so sight, sound, smell, touch, feel, and taste. Though we don't always use all five of them, hopefully you will not be tasting anything in the lab. And we use instruments to perform tasks that we can't do with our senses alone. And in this way we explore the unknown concepts of the world around us. Science is done by everyone every day, and we do this to develop new knowledge, to answer those questions that we have. And sometimes people don't actually realize that science is taking place, but when you're cooking in the kitchen, that is chemistry. Even just existing, there are hundreds of reactions that occur inside your body every single day to keep you alive. So when we have these questions, we use the scientific method to answer it, and this is just our way of collecting data, and it's a general process that has been developed, and we are going to talk about that a little bit today and the steps involved. So the first thing you need to know about the scientific method is that there are actually many expressions of the scientific method. Sometimes it is listed in 7 steps. I've seen it in 5, I've seen it in 3, and I've seen it in 10. Some expressions are a little bit more specific than others, but they all generally have the same process involved. So you always start with observation, and this is the information that you gather from your senses. And after that, the steps aren't always done in order, but we tend to see that after there's an observation, there's usually a problem that's identified, followed by a hypothesis. Then you will test your hypothesis. From that testing, you will get data. You will analyze the data to draw conclusions. And the last step is always to draw your conclusions and then share the information. In the scientific community, communication is the key. We would not be the society that we are today if we did not communicate both nationally and internationally with new scientific ideas. So let's take a look at the first step, which is observation. So the questions that we aim to answer using our observations is, why is it that way? It could be as simple as, why is the sky blue? And from there, you want to start to draw in other information that you can find. So it kind of starts the research process off. What do other people know about this, and how can I find out more? So at this point, you would go to find other people who have already done this experiment, and you would want to look at their findings. And even if their experiment failed, you could still get valuable information about either what not to do, or you could find information on how to improve on what they have already done. So this information that you collect in a scientific research paper, like what you would see with the science fair, this kind of gets condensed into like a 2 or 3 sentence purpose, just a purpose for the experiment. But in a formal lab report, this becomes the introduction, which is typically several paragraphs long, and this would be where you would cite your outside information. After we have some background research and we've made our own observations, the next step is typically to form a hypothesis. And if I asked you what the definition of a hypothesis was, you would probably tell me that it is an educated guess. And while that is a basic definition, we want to look at the difference between a prediction, an inference, and a hypothesis. So a prediction really is an educated guess. This is a guess of what will happen based on your prior experience. Okay, so something like, I know if I throw this ball up in the air that it's going to come back down because I've seen things fall out of the sky, like rain. Okay, so based on a prior experience. An inference is an explanation based on evidence and reasoning. So I know that this ball is going to come back down when I throw it up because of gravity. Okay, I'm explaining why it comes back down. And a hypothesis is a testable explanation. And this is what will guide your experiment and how you will form your experimental design. So let's look at an example. So you can see in the two pictures here, we're going to look at plants. In this case, it's form. And my observation might be that some plants are taller than others. Okay, so then I go and I do my research and I find out that soybeans can act as a fertilizer. So from there I want to make my hypothesis, which is that corn with fertilizer will grow taller than corn without fertilizer. So the key to this is that it is testable, and we can test this under controlled conditions. Alright, so when we're looking at designing an experiment, controlled conditions or controlled experiment really more so becomes a comparison between two groups. There's typically only one difference between the groups. When you have more than one difference, it gets difficult to tell which difference is producing your results. So our first group will be our control group. And in this case, it will receive what normal plants receive, which is sunlight, water, and soil. Our experimental group will receive the exact same conditions, but they will also receive fertilizer, in this case, the soybean fertilizer. Okay, and we're not necessarily going to give this to a whole field of corn. We're going to use a sample group. So a sample group is representative of the entire population. So for example, if we wanted to know, what the average number of texts that an 8th grader sends a day, we would not survey every single 8th grader at our school. We would pick 2 or 3 students from each class, and they would represent the 8th grade as a whole. In our experiment, we have 3 types of variables, and I apologize, I misspelled variables, and these are just factors that can change. So the first is a constant variable. This is also sometimes called the control variable, and this is what stays the same for both groups. So in this case, both groups will receive water, sunlight, and soil. The independent variable is the one difference between them. Alright, so this will be the fertilizer. This is what I, the experimenter, am changing. The dependent variable is what changes as a result of adding the independent variable. What can I measure to prove that fertilizer does or does not have an effect on plant height, on how tall they grow? So in this case, we want to know how tall. We would measure the plant height, which would be our dependent variable. So now that we've established our variables, that kind of concludes the experimental design phase, and we can move on to implementing our experiment and collecting data. So when you collect data, it's often raw or unprocessed measurements, meaning that you're basically just left with a bunch of numbers typically, or sometimes descriptions. And there are 2 types of data that you can get. The first type is qualitative, so kind of like the word quality. It is descriptive in nature. It typically uses comparisons, sometimes pictures, and an example of this would be that the height of the corn with fertilizer was taller than the control group. Okay, but typically we like to go for something a little more specific, and we like to support with quantitative data. Quantitative coming from the word quantity, so it typically has to deal with a numerical value. And you also typically get this numerical value by using instruments, even if it's as simple as measuring with a ruler or a yardstick. So in this case, our data would be that the height of the corn with fertilizer averaged 2 centimeters taller than the control group. I'm still saying the same thing, that the plant is taller, but by using quantitative data or numbers, I'm being more specific. Once you've collected your data, you move on to the analysis phase, where you get to interpret what your data means. And this becomes the results section of a lab report. And it's basically just an organization of your data. So you would take that data that you get and you would put it into tables, graphs, or some other sort of visual representation. So here are some examples of different types of graphs or visual representations. You can take a look at these. They're all used for different reasons. Bar graphs you might see a lot of in biology. Pie graphs you'll see more in math. It deals with percentages and fractions. In physical science, the number one graph that we will use will be the line graph. We like the line graph because it shows change over time, and we can measure that change over time by measuring the slope of the line or determining the equation of the line, which we will do. So this becomes especially handy for measuring things like velocity and acceleration. So once you have interpreted your data, this is where you get to draw a conclusion, and basically you get to explain your data and you get to share what you have found. So you want to give a general explanation of why your numbers or your graphs look the way that it does, and then you always want to go back to the hypothesis. So does your data support the hypothesis, or does it refute the hypothesis or go against the hypothesis? So this is a very important step because the hypothesis should be the basis of your experiment. But it's very important to note that if your data does not support your hypothesis, that does not necessarily mean that you messed up and you have to do the whole thing over again. You can get just as much information from a failed experiment or an experiment that does not support the hypothesis than one that succeeds. So just keep that in mind. The next thing you want to identify is any errors. So if you're using an instrument that maybe wasn't calibrated correctly, or anything that might cause a discrepancy in your data or cause it to look very inconsistent, you want to identify and explain those errors. And last but not least, you want to provide the next step, which is to improve upon the experiment, or even if it's to take it in a new direction, to identify the direction that you will go in from here. The number one thing about the scientific method is that it's never really over. You always start back over again. So here is an overview of the scientific method. I included a different graphic organizer than you saw earlier so that you can see what I was talking about sometimes it's in 4 steps like earlier. This one has more than 4 steps. So it is sometimes different, but overall the scientific method is used to test explanations, refine ideas, test them again, and of course always communicate and share what happened.